Well, what I'm going to try to do, and try is the operative word, is to make a really rapid tour of the kind of current state of the knowledge of the fossil record for the origin of the modern phyla. And before I do that, I want to discuss a few very important terms that really help clarify the discussion of the fossil record, or really evolution in general and the relationship of organisms. And so here we have a typical branching diagram. But what you see on the right in the shaded blue area is something called a crown group. And a crown group is defined as all of the living representatives of a particular modern taxon. So it could be a family or an order, or it doesn't matter what. And so all those living organisms that are placed within that category and their common ancestor and all the descendants of the common ancestor of those living organisms are all placed within the crown group. And all those organisms share all the fundamental characteristics that define that group. Then there's a stem group. And this term recognizes the fact that the features that characterize a particular taxonomic group don't appear all at once, suddenly in the fossil record, but they appear piecemeal. They appear in a stepwise fashion. So what do you do with the organisms that are kind of part mollusks or part arthropods or part chordates? They have some of the features of those taxonomic categories, but not all. Those are called stem groups, okay? So these are essentially transitional forms. They're transitional to the crown group. They have possessed some of the crown group features, but not all of them. And then you have, shown over far on the left, there are sister groups. And a sister group is a group that is most closely related to a particular, what's called a total group, the stem and crown group. It's more closely related to that than it is to anything else. And it's an extinct group that is closely related to the group of interest, more closely related to that than any other group, but it doesn't share enough characteristics to be placed in a stem group. The other thing I want to look at before we start going through the fossil record is this time diagram. And the reason I have this up is when you hear about the Cambrian talked about, or the Cambrian explosion, I think the common perception is that this is a really short period of time. It depends on how you define it. If you define the Cambrian explosion as the time interval during which modern phyla appeared, so beginning with the sponges and ending with actually the last appearing modern skeletonized group, which is the bryozoans. First sponges appear about 580 million years ago. The first bryozoans about 490 million years ago. That's 90 million years. Okay? The time since the extinction of the dinosaurs is 65 million years. The Cambrian itself, if you just look at only the Cambrian and ignore the late Precambrian record, goes from about 535 million years to 490 million years. 50 million years or so for just one period. Just the Cambrian itself is almost as long as the time since the extinction of the dinosaurs. So we're talking about very, very long periods of time. So I'm going to start with, kind of move up the taxonomic hierarchy, starting with the sponges. And currently, the oldest known body fossils for sponges appear about 580 million years ago in China. This is called the Ediacaran, the latest Precambrian. And this is from a paper describing those. And these are phosphatites. These are very tiny fossils preserved by phosphate. And there's some body preservation as well as the spicules. They're characteristic of sponges. So we have actual body fossils going back 580 million years. And based on biomarkers, particularly the biochemical remnants of steroids that are characteristic of sponges. From those biomarkers, we can extend that record back to 700 million years. And we have later in the Ediacaran, we have other body fossils of sponges representing different subgroups within the sponges, such as these hexactinellid sponges or glass sponges. We also have cnidarians appear in the Ediacaran. This is still latest Precambrian. And cnidarians include things like jellyfish, sea anemones, corals, hydrozoans. Those are all belong to the cnidaria. And these are jellyfish-like organisms from the very early Ediacaran that probably were not polyps like we think of jellyfish today, but were probably attached to the seafloor in kind of an upside-down position. But they are at least jellyfish-like in their anatomy. We also have anthozoans. Anthozoans are the group that includes sea anemones and various corals and, again, hydrozoans. And these are fossils, again, just very recently described from the Neoproterozoic or the Ediacaran. These are very, very tiny. Almost all these fossils are very tiny. These are soft-bodied. Everything I've talked about so far is a soft-bodied organism. On the left, you see cross-sections of things that seem to be like what are called octocorals today, that have a 8-fold symmetry, and a zoantherian, see on the right, which tends to have more of a hexagonal symmetry. But these appear to be similar to those modern groups. And another thing that I think surprises people a lot, despite all the talk of the Cambrian as being when all the skeletonized, mineralized organisms appear in the fossil record, there's actually mineralized fossils that appear in the Precambrian. And these are some of them. Most of them are these tube-like structures that you see in most of the diagrams. The one on the left there, the branching tube-like structure, and then right next to it is a close-up of that. And you see it has these horizontal partitions in it. This is probably an anthozoan. Again, we don't know for sure. There's no soft body preservation. It's just a partitioned tube. But one likely possibility is an anthozoan. Also, there are various kinds of tubes, calcified tubes. As you see, these are, again, very tiny structures, but they're very, very abundant. We don't know what lived in them, whether they're worms or, again, we don't know what the maker was. Interestingly, some of those tubes are bored. They have holes bored through them by a predator. So not only did the organisms exist that made these tubes, but there was something around, again, we don't know what it was, that was capable of boring through those tubes to prey on the organisms that were living in the tubes. Another interesting thing, we don't know what it is, lower right-hand corner, again, very tiny, associated with algal mounds or stromatolites that were very common at this time. And these are kind of these little bell-shaped things that were probably some kind of a filter-feeding organism that we don't know what its biological affinity was. But the point is, by the end of the Precambrian, we have certainly sponges, we have certainly cnidarians, including jellyfish-like things, anemone-like things, and even coral-like organisms existing by the end of the Precambrian, as well as a variety of other things that we don't know what they are, that were leaving remnants. By the early Cambrian, we start getting some nice body fossils. This is the earliest preserved sea anemone, an anemone-like organism. Again, this is soft-bodied. It's preserved by phosphate. Very, very tiny. See, the scale bar there is 0.2 millimeters. So these things are only like a millimeter across. These are very, very tiny little polyps. One thing that characterizes the Ediacaran is really important as far as the transition from the Precambrian into the Cambrian is that the seafloor had a tremendous transformation. At the end of the latter part of the Precambrian, the seafloor almost everywhere was covered by an algal mat. They're called mat grounds. So virtually everywhere, the seafloor would have been covered by these sticky, mucilaginous algal mats. And all the organisms that were living at the time, all the multicellular organisms that were living at the time, were living on those mats. They were feeding on those mats or using them as a place to attach, okay? And so a lot, see on the right, what are called Vendian fossils, which are what you usually see illustrated for the E.D. Cairn, these really spectacular, peculiar branching, kind of feather-like looking organisms. We really don't know what they are. There's a huge amount of argument and debate about what their biological affinities are. They may be somewhat related to living phyla, or they may be completely unrelated. But the point is that the surface were covered by these mats, and that affected the kind of ecology that it could exist at the time. What you see at the left is one of these preserved mats they called elephant skin. You see it out bedding plane exposure, and it's all kind of crinkled and wrinkled because of these algal mats. But in addition to those Vendian organisms, we don't really know what they are. There were other things that had much more modern-looking biological affinities. There are things that are bilaterians. Bilaterians means they're bilaterally symmetric. They have a head and a tail. Again, we don't know. There's some argument as to whether these fit into modern groups or not. But the point is we have something, it's a fairly complicated organism. It's bilaterally symmetric. It's motile, has a head and a tail. These are other examples of those. One reason why we know they're motile is they left traces. What you see at the bottom right is something called Yorgia, and on the left side is a bedding plane surface that has all these impressions of Yorgia as it kind of moved along the surface, moved across this algal mat surface, probably feeding and absorbing the algae as a food source. But also among these E.D. Cairn, again, we're still talking Precambrian here. We haven't gotten to the Cambrian yet. Are probably the most first widely recognized stem mollusk. Remember, stem means it has some of the features of modern mollusks, but not all. So it has some features that we recognize to place it in the phylum of mollusks, but not all of them. And what you see here is Kimberella at the top. As David mentioned earlier yesterday in his talk, has apparently a muscular foot. It also has some kind of a rasping feeding structure that leaves scratches behind. So you see it trailing behind the structure, or this kind of scratch marks on the algal mat. So this is something that was feeding on the algae. So this is a stem mollusk. Another thing that we have in the late Precambrian are burrows. But again, we don't know what the burrow makers are, but they indicate that there is a moderate diversity of organisms that were able to move through the sediment. These typically are mining out below these algal mats and feeding below the algal mats. Some kind of worm-like organisms, we don't know what they are because we don't have body fossils. All we have are their traces, but we know they're there, okay? And again, this is all before the advent of the Cambrian. Again, same thing. Another very common late Precambrian trace fossil. When you hit the bottom of the Cambrian, actually what defines the bottom of the Cambrian is this particular burrow, Tropichnus pedum. And this signals a dramatic change. And what happens is you suddenly get more and more burrowing organisms. And what they're doing is they're disrupting these mats. And also have things like Kimberella already. We know there's things that are feeding on the mats. And so now the mats get broken up. Now the bottom goes from this flat algae mat coated mat ground to soupy sediment. And now the organisms have to adapt to a completely different environment. They now have a soupy, soft, muddy substrate they have to adapt to, not this nice, stable algal mat. And that's because of the advent of burrowers. One of the first of those are parapulid worms. and they are probably the ones that made that Tropichnus pedum burrow. And Priapula has become really common worm phyla, so really common worm phyla in the Cambrian. Also, beginning of the Cambrian, you have something called paleoscelesids, extinct group of worms that are probably transitional between Pirapulids and a group I'm going to talk about in a little bit called lobopods, which actually lead to the arthropods. Mollusks, I won't spend a lot of time on this. We already said stem mollusks were already around in the Precambrian. By the early Cambrian, we have some true crown group mollusks. The earliest crown group mollusk that we know of is in the lower left-hand corner there, Odontogryphus. It has a radula. These right in the center at the top there, a rasping feeding structure that's characteristic, one of the defining characteristics of mollusks. At the top, you have a little later in the Cambrian, one of the early annelid worms, polychaete annelid worms, which have these characteristic chetae, kind of chitinous structures covering their bodies. But what's interesting is in the earliest Cambrian, you have a variety, very wide diversity of organisms, the wawaxids, the halcarids, and others, which have both mollusk and annelid characteristics in the same organisms. So these are sister groups, okay? These are sister groups to the mollusks and annelids that have some mollusk characteristics and some annelid characteristics. In other words, they represent a group of organisms that branched off before mollusks and annelids became distinct. So again, they're transitional. These are transitional forms between phyla, between annelids and mollusks. And then we have mollusks themselves. Again, the first true crown group mollusks appear in the earliest Cambrian and quickly diversified in periods called the small shellies. All these tiny little diverse skeletal structures appear in rocks, not at the base of the Cambrian, but a little above the base of the Cambrian. And one of the characteristic fossils that are in there are these little mollusk shells. And the halcyonellids are these little cap-shaped things. What's really cool, some of them become laterally compressed, weaken the shell along the dorsal side of the shell, and eventually those become two valves. And there's Fordilla is one of the earliest bivalves. So what we do is we see a beautiful set of transitions from those cap-shaped shells to the first bivalves. We also see good transitions between them and the first gastropods. Another thing, group that appears in these small shellies are things were called timodids. Nobody knew what they were. Those are just bits and pieces. They knew they had parts of something, but they didn't know how they fit together. And just recently, they found some that were attacked, so the various pieces were together. And they realized that these are actually stem brachiopods. Another group, important group of invertebrates, shelled invertebrates. And they began as these more conical tubular kinds of things with an opening at the bottom. And then they had a kind of a cap covering shell at the top. And at the lower right, you have something that's approaching the anatomy of a brachiopod shell, which you see here, which have two distinct valves. They have an opening in one valve that has a muscular structure that attaches it to the seafloor. Then lobopods. Lobopods I mentioned, I said I was get to because they are perhaps related to these paleoscelecid worms. Again, these were originally just known as these little tiny mineralized pieces until some really important discoveries in China and elsewhere recognized that they were parts of larger organisms, which were kind of caterpillar-like organisms with fleshy lobopod limbs. And here you see a variety, and they're incredibly diverse. And some of them become increasingly arthropod-like. What you see here is just a scheme phylogeny of the arthropods and the development of the characteristic arthropod anatomies from these lobopods. One thing that happens is they get a simple head appendages. They have eyes. Then they develop gills paired with their lobopod limbs. Then the lobopod limbs become mineralized and jointed in more arthropod fashion, and eventually add all the features that can be recognized as a modern arthropod. Here's one of the stem arthropods. This is like a step beyond the lobopods. It has both gills and jointed limbs. Anomalocharyd, one of the top predators of the Cambrian, which is again a stem arthropod. You can see the feeding structures on the head and the flaps there, which are the gills, and it had lobopod limbs, under these fleshy limbs underneath. So we have a really nice set of transitions going from lobopods to arthropods. Another important group, and that's because it's a group that we're closely related to, which are the echinoderms. Now you have chordates and echinoderms look like vastly different kinds of organisms. But they share some very important basic similarities. Echinoderms are things like starfish and echinoids and things like that, and crinoids. Well, again, one of the things about echinoderms is that they have a very unique skeleton made up of these calcite plates. No other phylum has them. And there wasn't really any understanding of where this anatomy came from, where the echinoderm anatomy came from, until this discovery, again in China, and it's a soft-bodied stem echinoderm. And it doesn't have the calcite plates characteristic of modern echinoderms, but has in-current and ex-current pores and other structures that place it close to that group. And if you look at the first echinoderms, they're really weird. These don't look like starfish and echinoids and echinoderm, or other modern echinoderms. They're these kind of sac-like or flat two-dimensional organisms that lay on the seafloor, covered now in these calcite plates. These are other early echinoderms. It's not until a little later in the Cambrian that they actually start showing this five-fold symmetry that we recognize as characteristic of echinoderms. Last group. Deuterostomes include both the echinoderms and the chordates, the group that we belong to. And this is a recently recognized fossil that appears to be a stem deuterostome. That is something that preceded both the echinoderms and the chordates. It's very primitive. It's just a sac-like body with a structure at the back, probably for locomotion, just a simple opening for the mouth, and a series of holes down the body as excurrent pores. So this is kind of like a sac with a tail, okay? And a little later, what you find is these, the B to the bottom there, and these are chordates. That is, they have a head, they have a tail, they have a series of gill slits running down their bodies. Their bodies are broken up into segments with muscle bundles. And we can recognize them as fitting into cephalochordates. The modern example of a cephalochordate is the lancelet at the top, sometimes called Amphioxus, or it used to be called Amphioxus. And then there's us. Earliest recognized vertebrate, later in the Cambrian. And this doesn't look much like us. It doesn't even have any bones or vertebrae, but yet it's a stem vertebra. How's that possible? Because actually vertebrates aren't defined by having bones, defined by things of their cranium, by aspects of their pericardial sac and muscle bundles and so forth. And this is one of the most primitive representatives of that group. So the point is, within this long time period, of several tens of millions of years, we not only have the appearance of most major modern phyla, but also their ancestors and the transitions that connect those various groups. So that's it. Thanks. Any questions? Yeah. Well, yeah, there's actually been papers that are just focused on the evolution of symmetry. And I'm not too familiar with that literature. But the echinoderms are a really interesting example because, the question was about the origin of symmetry and difference between hexagonal or pentameral symmetry and how those various symmetries developed. And within the echinoderms, for example, the earliest forms were completely asymmetrical. They had no symmetry at all. They were completely irregular. Some were just globular structures. Some were spiral structures, all kinds of bizarre morphologies. And it was only later that those symmetries, either bilateral symmetries or pentameral symmetries, kind of became the norm. So at least in that case, the symmetry was something that actually, characteristic of that group actually appeared after the group had been in existence for quite a period of time. So what it was that drove that particular success for those particular symmetries versus being asymmetrical or having different symmetries, I don't know. Bilateral symmetry is obviously something that's valuable for something that's motile. So actually the bilateral asymmetric echinoderms were motile echinoderms. So motility, directional motility kind of demands a bilateral symmetry.
Have talked so much about biology as a geologist, and I'm impressed with that. But you mentioned China. Where else is there any other place where?
Yeah, there have been important discoveries throughout the world and in Europe and Canada and other places. The reason China is so important, and it's almost important across the board in paleontology. Doesn't matter whether you're doing dinosaurs or invertebrates or plants. It hardly matters what you're looking at. China is really at the forefront of new paleontology discoveries. The reason that is, it's a big place. And in the 1920s, China closed down. There had been some exploration in China, international exploration in China before that, basically got shut down for a number of decades, didn't open up until the 1990s. So imagine how big China is. Let's say all of the United States or all of North America was completely unknown. And then you open it up, and not only was it opened up, but China suddenly realized this was a source of national prestige. This is a way of getting on the international map. So they'd actually go into, this is what I understand from people that have been there, not myself, but they'd actually go into, an incoming college class and say, you're a echinoderm specialist. You. And they have like more paleontologists per capita than we do in the US. I mean, they just like saturated their country with paleontologists. So it's not, you take a fertile kind of unexplored nation and you dump thousands of paleontologists on it, and what do you get? An unbelievable number of discoveries. And so it's really exploded our understanding in virtually every area of paleontology.
Why explosion in quotation
marks? The reason it's in quotation marks is, although it's a very commonly used term even within the scientific community, you'll see it in professional public papers, that I think it's very misleading as far as public understanding of what's going on. And as I said, even if you take the narrowest possible view of what the Cambrian explosion is, you're still talking maybe about 30 million years. Even if you lop off the late appearing phyla and you lop off the early appearing ones and you just concentrate on some of the ones in the middle, is still half the time since the extinction of the dinosaurs. Now, if you want to call that an explosion, yeah, in the whole spectrum of, 3.5 billion year history of life on Earth, yeah, it is an explosion from that perspective. But from typical person on the street, explosion implies something instantaneous, very sudden. This is something that took place over tens of millions of years. Yeah. Yeah. Right. Again, it was the appearance of organisms that could both consume the mats and what we call biointegrate, stir them up, break them up. And once you had organisms that did that, then it changed the whole kind of ecological system. And you both now have organisms that can penetrate down. So you now have, before everything was two-dimensional, ecology was two-dimensional. Now it gets three-dimensional, but it can go down. And you start putting nutrients up into the water column. So for the first time, before that, you didn't have any planktonic organisms. Everything was on the surface. So now things swimming, things in the water column, there's food sources in the water column, and there's food sources in the sediment, and now you're making it three-dimensional. And that is coincident with the explosion of body plans and diversity of feeding structures and so forth, because some of the resources were much more diverse.